Hi, good morning, everyone, and I appreciate the invitation to speak. For the next 15 minutes, I'm going to tell you about a project called ENIGMA and its relationship to global neuroscience. And the talk will be about discovering genomic loci that affect the speed of brain development and aging in worldwide populations. So just beginning with disclosures, I want to disclose consulting payments from Biogen and Kairos Venture Capital, but this is unrelated to the topic of this work. So the learning objectives for the talk are to learn how large-scale genetic analyses of biobanks can help us to understand brain diseases. And I'll explain how big data methods are being used to combine genomic data and brain scans on a global scale to answer urgent biomedical questions. And also we'll review some of the latest discoveries on the genetics of brain development and aging. So a little over 10 years ago, our consortium called ENIGMA began to publish some of the largest neuroimaging studies in the world, focusing on 11 brain disorders. We were interested in how diseases affect the brain worldwide, what are the genetic factors that affect the brain. And this led to an analysis of brain scans and genetic data from now a little over 100,000 people from 45 countries, with a little over 2,000 scientists examining Parkinson's disease, epilepsy, bipolar major depression, the major neurodevelopmental disorders of OCD, ADHD, and autism spectrum disorder, and many other conditions that I'll tell you a little bit about. So with brain imaging, you could examine the structure of the brain with anatomical MRI, its molecular composition using spectroscopy, its functional activation and synchrony using functional MRI. But we don't often think of relating these observations about the brain to omics data or genetics data, and I'll tell you a little bit about that in a minute. So collecting large quantities of data gives us three advantages. One is the sheer statistical power that you can obtain to tackle new kinds of questions. So if we were interested in what are the genetic drivers of brain development and aging, it'd be very hard to solve without very large data sets. There's also a lot of efficiency in using massive distributed computing across the world. We can do novel mathematics like deep learning or cooperative machine learning using the computer infrastructure of different institutions. And also the team science aspect. Many of the questions we lack individually the knowledge to solve them, but collectively we can do so. So here's where the brain scans are collected. They're all over the world. Many different scanners collecting brain imaging data from patients at the moment. And this has led to really quite in-depth studies of different brain disorders. This is a map of the regions where there is structural gray matter thinning in each of the disorders that's shown here. And you can see how characteristic patterns of brain abnormalities are associated with different conditions. And you can also get a t-shirt with the data on. So one interesting question is whether genetic variation at the DNA level is related to some of these brain imaging measures. The psychiatric genetics community often collects genome-wide association data from patients and controls. And here I'm going to explain a little bit about how to explain single nucleotide polymorphism variance effects on brain scans. So in this study, in the journal Nature a few years ago, we examined the volume of various subcortical structures of the brain on MRI, many of them implicated in neurological and psychiatric diseases. And we asked the question, are there common variants in the general population that are associated with the size of these structures? And the answer is yes. So the method is that you march along the genome and examine the genetic code at different points. And if there's a genetic marker that's associated with, in this case, the size of a brain structure, but it could also be its activation or other characteristics, you measure its statistical significance. So in ENIGMA, the members compute brain measures from different kinds of brain scans. Some of them are anatomical MRI, others like diffusion tensor imaging, examine tissue microstructure and its integrity, could also be applied to functional activation. And we test associations between these brain measures and over 1 million single nucleotide polymorphisms in the genome. And in aggregating data from many sites, we meta-analyze it. So we combine the effects across sites with a vote that depends on the sample size at that site. So this is beginning to be very interesting. We discovered 6 genetic loci that are associated with the volume of the hippocampus, the brain's major memory and learning system. You can look at the effects of famous genes, ApoE4, which is a risk haplotype for Alzheimer's disease. Each copy of E4 increases your risk for Alzheimer's by a factor of 3. You can see that carriers of E4 have a shift in hippocampal volume that's minimal early in life and about 2 standard deviations later in life. And this is a very tangible effect on the brain that likely produces the conditions that we just mentioned. Now, if you apply this to different structures, these are different components of the basal ganglia, the hippocampus, the brainstem that are shown here. You'll see different genetic markers associated with the volumes of different structures. And as you proceed, I mean, these studies are really becoming very, very well powered. You'll see that you could create a polygenic risk score, or essentially an estimate based on a person's genetic code of the volumes for different brain structures that you see here. And you can see that there is a reasonable degree of fitting. It accounts for now about 4 or 5% of the variance in these brain structures. It was simply to measure these markers that I've already mentioned. Polygenic prediction from the genome of brain morphometry is getting better for several key brain structures. As I just mentioned, we can account for about 5% of the variance. This is from known genetic markers, ones that you can specify, and this tends to increase with sample size. So as you travel around the human cerebral cortex, you'll see different functional territories. The genetic markers associated with each of these are partially overlapping and also partially distinct. And so if you look at this very interesting paper in Science from earlier this year, you'll see a whole host of genetic markers that are associated with this surface area or the thickness of different parts of the brain. There's hotspots in certain areas that affect the depth of the sulci, certain genomic loci that affect many, many different structures all at once. And you can cluster them. You can say, well, tell me what is the genetic overlap between the markers that influence one region of the brain and the others. And you'll see there's a modular genetic organization to the brain. There are certain clusters where the genetic determinants for different structural units overlap. And also there's an overlap between the genetic determinants of the brain structure and loci that put us at risk for conditions such as Parkinson's and Alzheimer's disease, also major depression, ADHD, and insomnia. And we're very interested in localizing in the brain where those disease risk variants exert their effects. So you'll see some of these types of maps here. Now moving to brain function, we can also assess the brain's functional synchrony using methods such as EEG. And by spectral analysis of these different recordings, we can do the same thing. We can identify functional genomic loci that are associated with different aspects of functional brain activity. This paper by Dirk Smit and the EEG group in ENIGMA identified a number of genetic markers associated with different frequency bands of brain activity. The GABRA2 gene has a variant, a common variant that affects beta power in the brain. This was a known genetic marker associated with alcoholism and epilepsy, but it's not clear why. And now I think there's some interest in pursuing in brain scans or brain synchrony maps what the effects of various neurological and psychiatric disease risk loci might be. Now, we talked a little bit about predicting the structure of the brain from your genomic markers. What about the future? So this is a study by Rachel Brower and Hilke Hulshoff-Poll where they essentially measured the rate of change in longitudinal MRI scans or the rate of loss of hippocampal volume, or in this case, lateral ventricular expansion. And they did this for all of the different structures that you can measure in the brain. And they asked, what are the genetic markers that are associated with the speed or the rate of changes in these structures? And you can imagine that this would be very interesting clinically to understand what the polygenic risk might be for future degeneration. And they found a whole host of markers in the genome that some of them are age independent in their effects. You can see an age independent effect of this polymorphism in the G protein coupled receptor 139. This throughout life seems to influence the rate of ventricular expansion. You can also see markers that are independent and dependent of age in exerting their effects. You can see the effect of ApoE that we just mentioned tends to have relatively little effect early in life and then a much greater effect later. So I think looking at these for different structures will begin to give us an understanding of the genetic mosaic of markers that influence the rate of brain development and degeneration. But can we be more analytically sophisticated? So obviously with any code, in particular the genetic code, understanding the principles, its multivariate organization, its covariance and interactions with other omics markers would be beneficial. And there's a number of methods that are being tried in ENIGMA and in imaging genetics generally that essentially combine multiple loci in the genome, potentially with other functional genomics data, to predict multiple loci or multiple signals in a brain image, or it could be multiple images. Some of these, such as sparse CCA, are very effective. They're simple linear algebra. Others, such as these methods developed by Professor Li Shen, constrain the search for either druggable genomic pathways that might be of interest to pharmaceutical companies, or known functional genetic units to guide the search in all of this genetic data. So just to wrap up, since 2009, our consortium ENIGMA has been involved in the quest to discover genetic loci that influence brain structure, function, and even the speed of and rate of brain development and aging. And you can imagine the value of a personalized polygenic risk score for your speed of aging, both for prognosis and to make adjustments in clinical trials. We're beginning to understand how very dangerous common variants in risk genes for Alzheimer's and Parkinson's, such as ApoE or the MAPT gene, affect different aspects of brain structure and function in large-scale biobanks. And also, just want to emphasize that scientists from over 45 countries are cooperating in this quest. So this offers greater power, more efficient computing, more expertise, and also, as is very important in genetics, understanding the generalization from different ancestries of the findings. I think in the future, we'll see a lot more work on deep learning and artificial intelligence, even on whole genome sequences to detect causal loci in the genome that influence brain and imaging traits. And just as one example, we're leading a new initiative called AI for AD, the Artificial Intelligence for Alzheimer's Disease Consortium, that attempts to use deep learning methods to unearth features in the genome that are associated with disease outcomes in Alzheimer's. And also this can be used by linkage to functional genomics databases for drug repurposing, identifying new targets in whole genome sequences. So a big thank you to all of the people in ENIGMA that contributed to this work. Huge thank you to the organizers for inviting me to speak today. And if you're interested, you can look on the ENIGMA website, you can join in with these efforts. If you're a geneticist or a mathematician or a computational biologist, you're very welcome to get in touch, and we'll try and look for interesting partnerships between this work and the other work going on. So thank you so much. Just want to acknowledge the funding for this work. This talk was recorded because it's 5 a.m. in Los Angeles at the moment, but I'd be very welcome and happy to answer questions in the chat later today. So thanks so much for your attention. Thank you.